Okay, let's finish up chapter 11. Yes, I adore Mendel. He, as I mentioned earlier, probably is one of the smartest people that ever walked the face of the planet. I don't disagree with that. However, Mendel had a very, very simple subject genetically when he chose the pea plant. Okay? Had he chosen any other subject to study, we probably wouldn't be talking about him, okay? Because Mendel's plants were so simple. Real-life genetics is much more complex than Mendel could have ever imagined. So let's just talk about a survey of topics that are not Mendelian. When I say Mendelian genetics, I'm talking about true dominance and true recessiveness. Okay, there are some things out there that their inheritance of traits is not like Mendel's pea plants. Okay, there's something out there called codominance. There are some organisms whose traits are codominant. Okay, co means together, like the word cohabitate means to live together. Codominant means that the alleles are expressed together, okay? Obviously, if you have homozygotes, that's not an issue, okay? But if you have an individual in a specimen that has codominance, if it's a heterozygote, that means that both of those alleles are going to be expressed. This is when more than one allele is expressed. So if you have a heterozygote, you're going to have both of those alleles expressed in that individual, okay? For instance, is anyone here familiar with cattle or horses? Horses? Have you ever heard of a roan? R-O-A-N? What's a roan? Anybody know? R-O-A-N? In cows, if you've got a red cow and a white cow, if it were Mendelian, you would think if you made that cross and both were homozygous, that your offspring would be all red, okay? Well, red and white are not dominant and recessive. They're codominant in cattle, which means when you have a heterozygote that has a red allele and a white allele, you get an individual offspring that has some individual hairs that are red and some individual hairs that are white. So both of those are expressed, and they look something like that. Camellias also do the same thing. Sometimes roans from a distance, because they have red hairs and white hairs, sometimes they look pink. Okay, so that's a roan. There is something called multiple allelic traits. And this is just what it says, multiple alleles, okay? Again, Mendel's pea plants were so simple. Every trait, height, color, etc., only had two alleles. If it was color of flowers, it was purple and white. If it was color of peas, it was green and yellow. If it was height, it was short and tall. So there were only two alleles, which made it a whole lot easier to try to understand what was going on. In most things, there's more than two alleles in the population, okay? For instance, human blood type. There's three alleles for human blood type. There's A and B and O. That's right. A and B are both dominant to O. A and B are codominant to each other. So let's talk about that for just a moment. There's three alleles for blood type. There's A, B, and O. An individual person only has two of those. Okay? So an individual person could have two A's, two B's, two O's. So obviously their blood type would be A or B or O, right? Okay? A and B are both dominant to O, so an individual could have AO or BO, okay? If an individual has AO, then their blood type would be A, because A is dominant over O. An individual that has BO would have B. But if an individual has both alleles A and B, those are codominant to each other. What does that mean? That means they're both expressed. Co-dominant, they're expressed together. So that individual would have AB blood because both are expressed. Okay? There is something called incomplete dominance. Okay? Incomplete means that neither allele is completely dominant. Okay? If you have a cross between two different phenotypes, you get a third phenotype that's totally different and appears to be blending of the two. Okay? So if you've got a red, these are 4 o'clock flowers. Anybody ever heard of 4 o'clocks? Yeah, they're really pretty, aren't they? Why do they call them 4 o'clocks? They bloom at 4 o'clock. They bloom in the afternoon, yes. Very pretty. But they have incomplete dominance, okay? So if you've got a homozygous red and a homozygous white and you cross them, you get an offspring that's pink, okay? So in looking at this, let's think about that for a moment. Is this blending, really? Think about that for a moment. Do you guys remember when we talked about blending in the very beginning, that old theory that was popular when Mendel was doing his work, and we talked about you could blend something, and it's when you blend something, you get something new and different, and you can't get the originals back out, right? That's blending. Well, if we look at this cross of 4 o'clocks, we have a homozygous red and a homozygous white, okay? We can't use big R and little r because neither is dominant over the other, okay? So instead of using big R and little r, we're going to use R1 and R2, okay? R1 stands for red, R2 stands for white. So we have an R1, which would be a homozygous red. We have an R2, which would be a homozygous white. When you cross them, the offspring gets an R1 and an R2, and that offspring appears pink, okay? But what would happen if Mendel had used 4 o'clocks? Think about that for a minute. If he had, instead of pea plants, if he had used 4 o'clocks and done this and got pink, he'd have said, well, yep, they're right. Okay, that's blending, okay? But right now, on your notes, I want you to, if this had been Mendel's F1 right here, I want you to cross 2 of those. So your cross is going to be R1, R2, crossed with another R1, R2. And tell me what you get. R1, R2, crossed with R1, R2. What do you get? When you cross R1, R2 with another R1, R2. You get 1 R1, R1, which Trent says is red. You get 2 R1, R2s, which are pink. Is that right? Yeah. And you get 1 R2, R2, which is white. So you got the red and the white back out. So it's really not blending, right? Okay. Also in, there's your cross right there. Genes, we used to think a long time ago, there was a theory that one gene would produce one protein, which would cause a phenotype, okay? We now know that that's not true. We know that genes are pleitropic, okay? That genes affect more than one thing, okay? A lot of genes are pleitropic. For instance, let me give you an example, Marfan syndrome. And the way that we know what a gene does, think about it for a moment. How do you know what a gene does? Well, when that gene gets mutated and it's not functional, you can see the effects of that, right? So there's a disorder called Marfan that is a mutated gene on chromosome number 15. And when individuals inherit this mutation, it causes lots of problems for them. It causes long arms, feet, hands, etc. I'm not talking about just a little bit longer. These individuals have disproportionately long limbs. Okay, they also have heart problems, and they also have problems with their eyesight. But their appearance tends to look like this right here. That's an individual with Marfan's. It causes extremely long appendages. Some theories out there about whether or not Lincoln may have had Marfan's because he exhibited some of the characteristics of this. I don't know. Just a theory. Polygenetic inheritance. I wish we had a whole chapter on this because this is real life genetics. Mendel's traits were controlled by one gene. Height was controlled by a gene, which has 2 alleles, right? Color was controlled by one gene, 2 alleles. Most traits are polygenetic. That means that the trait is controlled by more than one gene. So more than 2 alleles are controlling the trait, okay? It's when a trait is governed by 2 or more sets of alleles. Anything that is a color in humans is polygenetic, okay? Eye color, skin color, hair color, these are all polygenetic traits. This is why I didn't like the color of eyes problem in the monohybrid crosses, because it's not relevant anymore. We now know that your eye color is controlled by at least 3 genes, that's 6 alleles, and they're on different chromosomes. They're not even on the same chromosomes. So think about that as far as trying to figure out the inheritance pattern when you've got crossing over and independent assortment. They're not even on the same chromosomes. So anything that's a color, height in humans is polygenetic. When you have a polygenetic trait that's controlled by more than 2 alleles, each allele is actually producing a product. Okay? So the more dominant alleles you have that are producing the product, the recessive alleles are not producing the product, the more dominant alleles you have, the more of that product you have, right? So let's think about it for a moment. Let's say your skin color is controlled by, let's just say hypothetically, 6 alleles. Okay, if your skin color is controlled by 6 alleles, it would look something like this. Don't write that down yet, just listen, okay? Do you even have to write it down? No? Okay, good. Let's say that your skin color is controlled by 6 alleles, okay? Everywhere there is a dominant allele, there's a product produced. So if someone has 6 dominant alleles, they're going to have the darkest skin, hair, eyes, etc. Okay, anything that's a color. Someone that has 5 dominant and 1 recessive is going to be a little bit lighter. Okay, think of it like food coloring, culinary. Okay, if I want to make red cookie icing for Valentine's Day, I need red. If I put 1 drop, it's going to be pink, right? If I put 2 drops, it's a little bit darker. 3 drops, it's a little bit darker. If I put 6 drops, it's red. Okay, so the more that you have that are dominant, the more pigment you have being produced, the darker the color, okay? I love the biology of skin color because the only difference between people of different colors is you have more pigment, okay? You take the skin off, which is kind of a gruesome thought. You take the skin off and we are all just alike, okay? You won't be able to tell the difference, okay? Do what? I've been many, many times. The Bodies exhibition is polymerized human bodies. Yes.
That was so awesome. They wouldn't let us take pictures or anything.
That didn't stop me.
Oh, I was like, man, click.
Yes. And at the end, they let you hold like damaged parts. Did you get to do that? You didn't do that?
They didn't
like,
let us do that.
Yes. Guys, these are plasmization, I think is the word they use, I can't remember, of human bodies, and it's like there's dozens of humans that have donated sometimes.
Skin and their like, what was it? The whole nervous system.
Yes, they acid wash the rest of the system off, and they display just the circulatory or just the nervous system. Yeah, we used to give bonus points for anatomy students to go. Yeah. Yep. Oh, the fetal room's terrible. Because when you see the fetal development and each example, you stop and you really realize that was a human and that was a human. And yeah. If you're ever in Atlanta, it is a wonderful thing to go see. The Bodies exhibition, Bodies exhibit, yeah. Okay, so basically what we're saying is each dominant allele has a quantitative effect. What is quantitative? You can measure it. You can count it. Yes, you can measure it. So if you've got a dominant allele, there's a little more. Another dominant allele, there's a little more. It has a quantitative effect on the phenotype. So the more dominant alleles, the darker the color, the taller the person, etc. Okay? So things that the effects are additive. Things that are polygenetic are things like skin color, height, eye color, etc. When you have a trait that's polygenetic, what you end up with in the population is a continuous variation where you've got most of the individuals in the population in that middle range with less individuals on the extremes, really, really light, really, really short, or really, really dark, really, really tall. Okay? Does that make sense? Most human genetic traits are polygenetic. That's why you can't really do your eye color with a monohybrid Punnett square. Okay, let's don't underestimate the effects of environment. Okay? Environment has a whole lot to do with your phenotype. First of all, you're born with a genotype, right? That's the coding that tells that zygote what your characteristics are going to be, what you develop into, right? Well, let me ask you a question. Let's say that you have a genotype. Wait, first of all, what's a really, really tall basketball player? Who? Shaquille O'Neal. Does everybody know him? Okay. Let's say, how tall is he? 7'3". Okay. Let me ask you a question. Would Shaquille O'Neal have been that tall if, let's say, he had not received proper nutrition as a child? Probably not. He probably would not have met that, his potential height, right? I mean, you would have to have nutrition to grow. So you got to stop and think. Environment has a lot to do with your phenotype in lots of different ways, and that's one of them. One of my favorite examples is this little Himalayan rabbit. And do y'all know what a Himalayan rabbit is? Yeah. I want one so bad. I'll trade you what's that thing back there? A chinchilla. I'll trade you a chinchilla for a Himalayan rabbit. Yes, he does. He's been by himself for a week. You know, I mean, bless his heart. Okay, so these rabbits have an enzyme. And remember, enzymes are proteins, right? And proteins are very sensitive to temperature and pH, right? We learned that. So these rabbits have an enzyme that codes for black fur. The enzyme produces a protein, which is a pigment, that codes for black fur. But this enzyme is denatured. What does that mean? That means it's not functional, right? At the core body temperature of that little rabbit. So where the rabbit's body is the warmest, the enzyme's not producing the black fur. It's only producing that black fur in the cooler regions of that rabbit's body, the peripheral parts, which would be the ears, the nose, the feet. Yes. Is that not the cutest thing you've ever seen? Look at that. Okay, that's a Himalayan rabbit. So theoretically, if you put an ice pack on this rabbit's back, you could cause it to have a black spot. Okay, isn't that cool? Yeah. because that enzyme is denatured at that temperature. A Siamese cat's the same way. So if you put a Siamese cat in the freezer, if it lives, yeah, okay. Don't do that. Please don't do that. Okay, these flowers, anybody familiar with these flowers? Hydrangeas. These are your grandmother's flowers, aren't they? Yes, everybody, yes. These are very old flowers. The bloom of their, yes, sir. It doesn't happen that fast. Really? But they get darker when he goes outside? I don't know. What kind of dog is he?
We think he's a pit bull Dalmatian. He's a
Oh, I don't know. I'll have to look into it. I'm sure it wouldn't happen that fast. No.
But no, like when he gets out,
Yeah. Yeah.
okay.
Yeah. It would take longer than that. Yeah. 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 Wow, guys, going back to the polygenetic and the color and everything, that's why babies' eyes are born, they're blue when they're born, because that gene hasn't started producing a product yet. So when they're born, the gene activates, starts expressing, and it takes a period of time for that pigment, which is the color. Think of it like paint pigment, okay? It takes a while for that pigment to build up. That's why babies' eyes are born blue, and then if they're going to turn brown, they will turn brown months later. I don't know. I'll do some research. That's interesting. Don't put it in the freezer, please. Don't put it in the freezer. Yes. Yes. Yeah. It takes a while for that pigment, that gene, to start producing a product. We call it gene expression. And guys, genes, this is kind of going into chapter 12, but genes turn on and off, okay? Remember, every cell has the whole recipe book. And some cells use subsets of recipes, chapters, okay, and other cells will use other genes or recipes. And throughout your life, this may change. Some cells may use some genes earlier in life and then change and use other genes later in your life, okay? And that's a huge area of study right now, is gene expression. How do we turn genes on and off? How do genes know when to start or what product to start producing? Okay? I think that's probably hormonal. Yeah, I think that's probably hormonal. But yeah, I know what you're talking about. And some women too. Okay, guys, the hydrangea, as you guys, this class is very talkative. Have you noticed? Yes. Yeah, I know. I love it too. Okay. Hydrangea's bloom is dependent on the pH of the soil. Okay. I never can remember. I think the higher the pH, which would be basic, is a blue bloom, and I think the lower pH is the reddish pink bloom. I'm not sure. Sometimes I get those confused. I'm not a horticulturist. Okay. Me either, but yeah. Okay. Are y'all ready to go into the rest of chapter 11? Anybody have any questions so far? Any questions? Oh, we're still on the rabbit. Okay. It's his body temperature, okay? He has to maintain homeostasis just like you do, okay? So he has a body temperature that he would have to maintain. So his core body temperature is not going to change. He may shiver more, you know, I mean, he may grow thicker for, yeah, yeah. I would have to look at, yeah. I don't think it would change his core color. Okay, it might make it darker. I mean, I don't know. I'd have to look into that. I want one.
So would they turn white in like normal?
Y'all are hilarious. No, it's going to be his core body temperature. Yeah,
Oh,
yeah. Okay, and I would have to look up what the differential is, how much colder it has to be, and how long it has to stay. Okay, 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 guys. Human genetic disorders, medical conditions caused by alleles that you inherit from your parents. Okay. First of all, let's go ahead and remind you that when someone has a genetic issue, and this pertains to every issue we've talked about, number problems, structure problems, when someone has a genetic issue that they are born with, that means that that had to come from the egg or the sperm that formed them, right? And they inherited it, and it was copied, that problem, whatever it is, was copied into every cell of their body. Okay? Because as we go through this, inevitably someone is going to say, is there a cure for it? Well, no, these disorders that we've talked about so far and continuing to talk about are as much a part of who they are as your eye color is to you. You can't change it. It's what you are. It's who you are. Okay? Okay, there are some things we can do to alleviate some of the symptoms of this, but of some of these things, but no, these things can't be cured. Okay, but we've talked about number problems, we've talked about structure problems. When we talk about human disorders and we're studying them, we're not Mendel, and these aren't pea plants, right? These are human beings. So when we study human genetics, we can't cross people and study their offspring. You know, people don't take too kindly to be crossed. And even if they did, you would only get one or two offspring to study, and that's not good. So when we study human genetics, we have to look at the past. We have to look backwards to see how things were passed or inherited. Okay? So we're going to start now by talking about disorders that are not number problems, they're not structure problems, they're actual coding problems. Okay? So when I say coding problems, I'm talking about these people have the right number of chromosomes, they've got the right structure, but you remember that your genes are a piece of DNA, it's a section of DNA, right? Well, remember we learned that DNA consists of bases, nitrogen bases, nucleotides, that are joined together. So and we said adenines go with thymines and cytosines go with guanines. Remember we said that A's go with T's and C's go with G's. Well, sometimes your cells get it wrong. Every time your cells divide, sometimes mistakes slip through and it changes the coding. Okay? Well, if those changes happen to be in an area of a gene, then you've got problems sometimes. Okay? So, when we study these that are passed down from parent to offspring, we use these things called pedigrees. Okay? A pedigree chart is what you used to call a family tree, and you probably did one in the 4th grade, and you probably traced eye color through your family, or sometimes skin shading or color or hair color, height, something like that. Okay? The pedigree chart is going to show you everybody in the family and how they are related to each other. And if we're tracing a disorder, it's going to show you the phenotypes of those individuals, who has the disorder and who doesn't. Okay? So pedigree charts shows all known phenotypes for an organism and its ancestors. So let's make sure we understand some things about a pedigree chart before we go forward with our study of human genetic disorders. Okay? First of all, on pedigree charts, obviously this is going to be generation 1 at the very top. The next generation is generation 2. The next is generation 3. Males are squares or rectangles, and females are always circles. Okay, and that's easy to remember because females have curves, right? So the females are circles. Usually you'll have a key right here that's going to show you who has the disorder and who doesn't. Usually the people who have the disorder are a darker shade of something. Okay, so the way that I would read this, when you have a horizontal line, that's a union between two individuals. The offspring of that union is illustrated with a vertical line, okay? So the way that I would read this is this male, the yellow square, was united with that female that in this illustration is appearing as half and half, and we'll talk about that in a moment. They produced two sons that, according to my key, are both affected with color blindness. This son mated with this female who is normal, and they produced two offspring, a normal male and this female that is half and half. Okay? Some of these genetic disorders that we're going to talk about, it's possible for you to have a normal phenotype. Okay? You're not affected by any problems or anything, but you actually are carrying a allele that could cause it. Okay? And so those would be called carriers. Okay? Is there any question about this so far? Generation 1, 2, and 3. Usually our individuals are numbered. Guys are squares, girls are circles, etc. Everybody good with it? Okay, let's talk for just a minute. Okay, listen up for just a minute. It's really important that you understand this to understand the rest of the chapter. There are some genes that the alleles, you know, a gene is a segment of a piece of DNA on homologous chromosomes, right? So every gene has two alleles. Here's my alleles, okay? Every gene has two alleles, right? There are some genes where the alleles don't have to work together, okay? They don't work together. So as long as you've got one good copy of a working allele, if the other one is not working, you're still okay. Okay? As long as you got one good one, you're still okay. Now this one's not working, right? So if you passed that one on to your child, and your child happened to inherit another allele in that gene that wasn't working, then your child would not have a working allele for that gene, right? So they would have a problem, a disorder. Okay? We call this allele the affected allele because something's wrong with it, right? So from now on, I'm going to call this an affected allele. Well, if you have an affected allele for that gene, you're still okay as long as you've got a good one, right? We refer to these affected alleles as being recessive because the presence didn't change your phenotype, right? The only time that the presence of an affected allele would change your phenotype in these genes would be if you got two of them, right? Are you with me? You sure? Okay. There are other genes, okay, where the alleles have to work together, okay? So you got to have two good ones. So what happens if you inherit a bad one? You've got a problem, right? For those genes, we refer to the affected allele as being dominant because the presence of that affected allele is going to change your phenotype. Okay? So these are dominant alleles. You have to have two good ones in those particular genes to be normal. Are you with me? You sure? Okay. These affected alleles, these problem alleles that have coding issues, can be both in the autosomes or the sex chromosomes. They can occur anywhere. Okay? Tonight we're going to talk about those that occur in the autosomes, and when we get to the sex chromosomes, we're going to talk about those that are on the X. We're not going to worry about Y alleles tonight. It gets confusing, okay? And we're only going to talk about those on the X that are recessive. Okay? So, affected alleles can occur on an autosome. That's any chromosome other than the sex chromosomes. They can occur on the sex chromosomes. And when they do, we call them X-linked, okay? They can be dominant, meaning that the presence of the allele causes you to have a disorder. Remember, those are the alleles that work together. So if you get a bad one, you've got the disorder, okay? Or they can be recessive, meaning that you have to have two defective or affected alleles to have the disorder, okay? Are you still with me? So tonight, what are we going to talk about? We're going to talk about autosomal recessive disorders. We're going to talk about autosomal dominant disorders, and we're going to talk about X-linked recessive. So we're going to talk about three different kinds of these, okay? Okay? I did, yes. Okay. Okay, we're going to start with autosomal recessive disorders. Now keep in mind, these types of disorders would occur in genes where the alleles do not have to work together. Okay? Remember, in these genes, as long as you get one good allele, you're good. Okay? So, we say that the normal allele is dominant. Okay? Because as long as you get one, it causes your phenotype to be normal. The affected allele is the recessive allele in these particular genes. Okay? So the normal allele is dominant. The recessive allele is affected. Okay? And again, these are on the autosome. These particular disorders we're going to talk about are on the autosomes, which would be chromosome pairs 1 through 22. Okay? So, what could your genotypes be? If an individual has two dominant alleles, they're normal, right? Obviously. If an individual has a dominant and a recessive allele, they're still normal. But now these individuals we would call carriers, okay? Because they're carrying an affected recessive allele. So if an individual gets two of these affected alleles that are recessive, they have the disorder, okay? So big A is not affected. Big A, little a, the heterozygote, is a carrier, and homozygous recessive for these genes would have the disorder, okay? Is everybody good? Okay, let's look at what a typical pedigree would look like and some of the characteristics that would accompany this type or this pattern of inheritance, okay? Okay, here's your key over here to the right. This is what I told you just then, okay? Two dominant alleles is unaffected. A dominant and a recessive allele is a carrier with an unaffected phenotype. And two recessive alleles would be affected with the disorder. Whatever we're talking about, that individual would have the disorder, okay? So one thing that we can summarize from this is that all normal individuals are normal by virtue of the fact that they have at least one dominant allele, right? So if an individual is normal, we know that they have to be big A question mark at least, right? Are you with me? Okay. So all normal people have at least one dominant allele, one big A. Okay, so let's look at our pedigree for just a moment. We have a female that's affected with whatever disorder we're tracing, who mated with a male that's normal. They have two daughters that are normal. Obviously, they are both carriers now, right? Because mom was affected. The only way you could be affected with an autosomal recessive disorder is to be a homozygous recessive, right? Homozygous recessive, okay? This daughter mated with a normal man, had a normal daughter. This daughter mated with a normal man and had a normal son, another normal son, and a normal daughter. Okay? Do y'all see something weird? It more than looks like it, doesn't it? This family tree has no limbs, okay? It's a stump, okay? Okay, yes, that is what this double line between those two individuals indicates. It indicates that they are related to each other, okay? Okay, so this carrier female mated with that carrier male, and they have a son that's affected, a daughter that's affected, and then another daughter that they don't know, okay? That's normal, okay? Okay, so let's look at, well, yeah, normal, yeah, that's relative. Okay, let's look at some of the characteristics, and these are important. You're going to need to know these, okay? Most affected children, right, have unaffected parents. Most affected children have unaffected parents. Okay, why? Because both parents were, that would be big A, little a, crossed with big A, little a. Those would both be carriers, right? And when you do the Punnett square for that, you have a 1 in 4 chance of having an affected child. Okay? So when two carriers mate, they have a 1 in 4 chance of having an affected child. Second characteristic is heterozygotes have an unaffected phenotype. Well, that's by definition of, we said it was the affected allele was recessive, right? So a heterozygote's going to have a normal phenotype. Two affected parents will always have affected children. What is that? That is little a, crossed with little a. And what's your Punnett square look like? All little a's. That's exactly right. Everything's little a, little a, little a, little a. So two affected parents will always have affected children. Okay? The next characteristic, close relatives who reproduce are more likely to have affected children. Those are your cousins right there. Okay? Guys, these disorders, autosomal recessive disorders, are the reason, recessive disorders in general, are the reason that societies have laws against mating with family members. You thought it was just because it was gross. Okay? No. These are the reason there are laws, okay? Because even if you're a carrier, if you mate within the general population, that kind of sounds promiscuous, doesn't it? If you mate within the general population, the chances of you mating with another carrier is going to be slim, okay? And even if you do, the chances of you having an infected child is 1 in 4, okay? However, if you've got one of these alleles in your family tree, or if you limit your gene pool, you know what limiting your gene pool is? And limiting your gene pool, guys, can be more than just family. I mean, this is kind of obvious right there. But you could actually limit your gene pool with ethnicity, religion. There are some of these disorders that are more predominant in certain religions. For instance, there's one that's more predominant in the Jewish religion because most Jews in America came from about the same area in Europe, and up until 50 years ago, Jews married Jews. Okay? They wouldn't think of marrying outside their religion. So all these people came to America and they just continued to mate within the same gene pool. And so one of these disorders shows up in the American Jewish population more than, as a proportion, more than the general population. Okay? So these are the reasons that we say if you mate with a family member, if there is one of these alleles in your family tree, the chances of those two recessive alleles coming together is greater than if you mated within the general population. Okay? And both males and females are affected with equal frequency because we're talking about autosomal disorders. Okay? These are on chromosomes 1 through 22. So let's take a look at some of them. Okay? The first one is called methemoglobinemia. Ever heard of it? Methemoglobinemia. It is harmless. First, let me give you some background. Hemoglobin, which is the protein in your red blood cell that carries the oxygen through your body. Okay, it attaches to oxygen. Hemoglobin naturally breaks down into something called methemoglobin. Okay, methemoglobin will not bind with oxygen. Normal individuals have the enzymes necessary to convert metheme back to hemoglobin, so everything's good. Okay? Individuals with methemoglobinemia lack that enzyme. So when their hemoglobin breaks down, it stays methemoglobin. Okay? Which means that they have less oxygen in their blood. They're blue people of Kentucky, exactly. It causes these individuals to have a blue tint to their skin. And I was just going to ask, have you ever heard of the blue people of Kentucky? They think that they probably had methemoglobinemia. This is harmless. They do have some symptoms. They do have some health issues, but it is harmless because the average lifespan of a red blood cell is about 120 days. So when hemoglobin breaks down to methemoglobin, it's not fatal because those blood cells are going to die anyway and be replaced by other blood cells. Okay? It's on number 22. Cystic fibrosis. Does anybody know somebody with cystic fibrosis? Can you share anything with us? He does. Is he doing okay? Yeah, it's tough. It really is. Yeah. It's a defective chloride ion channel on chromosome. The gene is on chromosome number 7. What this means for the individual to have a defective chloride ion channel is that anywhere there is a secretion, the secretion is more concentrated and thicker. Okay? So they have really thick mucus, really concentrated salty sweat. Anywhere there's a duct gland, a duct is a passageway, anywhere there's a duct gland, there's a tendency for those glands to get clogged. Okay? So you have lots of pancreas problems. The pancreas is a duct gland. Lots of lung problems. Again, your sweat glands are duct glands, so your sweat is really concentrated. Matter of fact, someone once told me that their grandmother had told their grandmother, okay, so this goes way back, that if your infant tasted salty, that if you actually kissed your baby and it tasted salty, that it could have cystic fibrosis. Now, guys, I'm not talking about a 2-year-old. All 2-year-olds taste salty, okay? If your infant tastes salty, then there could be a problem. Okay, it is the most common lethal genetic disease among Caucasians in the U.S. 1 in 20 are carriers. Okay, so you know this is a class of 35. Technically, 1.5 of you probably are carriers. 30, 35. How many people know somebody with cystic fibrosis? Raise your hand. Let me count. 1, 2, 3, 4, 5, and 6 in the class. Okay, so just to give you an idea, it gives you a reference. Okay, 1 in 200 newborns. Niemann-Pick is on chromosome number 11. It codes for something called acid sphingomyelinase. This is an enzyme that's supposed to break down a lipid. A lot of these disorders are faulty enzymes that are involved in metabolic pathways. So when you get an enzyme that's not working, the pathway doesn't complete and you get a buildup of products that are not supposed to be there. Affected individuals don't break down the lipid. It accumulates in the cells of liver, lymph nodes, and spleen, so these people have a lot of abdominal issues. Most severe cases, it builds up in the brain, causes mental retardation. This is Niemann-Pick. Phenylketonuria, PKU. How many people have ever heard of this? PKU, raise your hand. Got a few people. Anybody know somebody with this? I actually have had 2 students in my class over the years that have had this. Very interesting conversations with them. Okay, little background before we get, everybody needs to know this. Guys, everybody in this room, if you ever plan on having children, you need to know this, okay? And you two need to know it especially well, okay? Because you're going to be taking care of people. What type of nursing are you going into? ICU? Cardiac? Okay. Okay. Phenylketonuria is the inability to metabolize an amino acid called phenylalanine. If you guys remember from that chapter 3 that I've talked about for like 3 months now, phenylalanine is one of the 20 amino acids, okay? Basically, phenylalanine is in everything that's a protein. It's also in artificial sweeteners. If you have an artificial drink on your desk, you might see that. These people are born not able to metabolize this amino acid. So they are born normal, okay? The problem occurs when you start to feed them and you give them proteins that have phenylalanine in them, and they're not able to completely metabolize that. Again, you get a buildup of a product that's not supposed to be there, and it will result in mental retardation. Okay? So, basically, these people are normal until you feed them. And then, if you feed them, you don't know it, then you will cause these things to happen. So the key here is to identify these people at birth. This is why babies get, well, one of the reasons, babies get the little heel prick, okay, where they take the drop of blood. They're trying to identify, are they phenylketonuria? If they are, then you can't even have breast milk. You can't have formula. You can't feed them, okay? You have to get a special prescription for a formula that contains all of the other 19 amino acids but lacks phenylalanine. So they have to stay on this very special, expensive formula. And then when they start eating, they're basically on a fruit and vegetable low-protein diet, okay? I had a lady one time that had my class that had this, and to listen to her story was just amazing because she was in her 30s, probably low 40s. She had never had a hot dog, never had a hamburger, never tasted peanut butter, never had ice cream, okay? I know. But to listen to her tell about her life was really interesting. She had to take a protein shake, a very special prescribed protein shake every day to get the rest of her amino acids. Anyway, let's get it in your notes. Chromosome number 12. There's 12. There's the heel prick. Brain damage results, 1 in 10,000 to 25,000, males and females equally. And the key here is to have a low phenylalanine diet. And here it is right here. The closer to the target, the less phenylalanine. So you got juices, vegetables, fruits, and maybe some low-protein pastas and breads. As you go away, then those would be things that would be prohibited. Okay? She said that after again, remember it's affecting the nervous system. She said that after she was developed, okay, after she was grown, that they allowed her to start having more phenylalanine as long as she monitored it because she was not developing anymore. But she said, get this, let's back up for a minute. Her parents were normal, okay? This is autosomal recessive. She was the 1 in 4, right? So her parents were carriers, big A, little a, crossed with big A, little a. She was the 1 in 4. Her parents had 2 children. Both had phenylketonuria. Yeah, they hit 1 in 4 twice, okay? She told us about her sister who also had it. She was the good child, okay? She was the good child, and she was proud of it. And we asked her, one of my students, she talked for about 30 minutes, and one of my students was asking her all kinds of questions about, don't you want to taste it? And she said, no. She said, I do not ever want to taste the peanut butter or the ice cream. She said, because if I ever taste it, I'll know what I've missed. I'll always want it. If I ever taste it, I'll want it. Well, her sister, who also had it, was the bad sister. Okay? It's almost the good witch, and the bad witch in the Wizard of Oz. She said her sister would sneak off and get a hot dog. Okay? Now, I want you to think about that, parents. Can you imagine your child being bad, sneaking off and getting a hot dog? Usually we worry about them sneaking out and getting drunk, or something like that, or something bad. She would sneak out and go get a hot dog, okay? And she said they could tell when her sister had been bad because it affects the nervous system. And she said it was almost a bipolar kind of schizophrenia, that it would change her personality when her phenol levels got up. And as an adult, she had to monitor those levels. If she was going to have phenylalanine, kind of like monitoring of insulin with a diabetic, that if she was going to have phenylalanine, they would have to take blood and monitor that her levels didn't get too high. Very interesting to talk to. But she was on this special milk, she called it a milkshake. I dare think that it probably tasted like anything we would consider a milkshake. But anyway, she was on this phenol aid for the rest of her life. That's the only way she gets her proteins. Okay, other autosomal recessive disorders, you may have heard of some of these. Sickle cell, Tay-Sachs is the one that's in the Jewish religion. Albinism, thalassemia, Usher syndrome, Alpers. Some of these you may have heard of, some not, but there's lots of them. We've just done a survey of a few of them. Okay? Okay, autosomal dominant disorders. Okay, let's change hats. Okay? Remember, these are the genes in which the alleles have to work together. So you got to have two good ones, right? You have two good alleles to be normal. So if someone gets an affected allele or an allele that's affected, then it causes a change in their phenotype, right? You with me? The affected alleles cause a change in the phenotype. So in these particular genes, the affected alleles are dominant. Okay? So the affected allele is dominant. The recessive allele is the normal one. Okay? Are you with me? Come on, guys. You got to follow this because it's just the opposite. You get a bad one, you got the disorder. Individuals that have two dominant alleles definitely have the disorder. Individuals who have one dominant and one recessive allele have the disorder. Notice in autosomal dominant disorders, think about this for a minute, okay? There are no carriers because the presence of the affected allele causes you to have a disorder, so you can't carry it, you can't hide it. If you get the affected allele, you got the disorder, okay? You can't hide it. The individuals who are normal have the two recessive alleles. No carriers. Okay, so let's take a look at the pedigree and some characteristics of the pedigree. You got to understand the key to be able to understand what's going on, okay? So here's my pedigree, and there's your key. This is what I just said, okay? Two dominant alleles are affected. A dominant recessive is affected, no carriers. So basically, we're summarizing that by saying anybody that has the disorder has to have one dominant allele, right? You got to have at least one dominant allele to have the disorder. And those individuals that are unaffected have two recessive alleles. Okay? Okay, so we have on our pedigree two individuals that are heterozygotes at the very top. Heterozygotes are affected, remember, because the presence of the affected allele causes you to have the disorder. So those two people are affected. They have a son that's affected, a daughter that's affected, and two daughters that are normal. Look at that for a long time. Okay? The normal daughter mates with a normal man and has normal children. The affected son mates with a normal female and they have a normal female daughter. I guess all daughters would be female, huh? A normal daughter, affected daughter, and an affected son. Okay, so let's look at the characteristics of autosomal dominant. Affected children will usually have an affected parent. That's because of what I said a while ago. You can't hide it. In dominant disorders, autosomal dominant disorders, if a child has it, remember the presence of the dominant allele causes you to have the disorder. So if the child has it, you should be able to trace back. He had to get it from somebody who also has it. Okay? So there's the allele that's causing the disorder. It had to come from somebody, and whoever gave it to him also has the disorder. You can't hide it. Okay? Heterozygotes are affected. Two affected parents can produce an unaffected child. You might want to highlight that. People miss that on the test a lot. Two affected parents can produce an unaffected child. Guys, that's this right here, and that's why I left this Punnett square on the board over here. Two affected parents would be big A, little a, crossed with big A, little a, right? Both of those parents are affected. As long as the parents are heterozygotes, they have a 1 in 4 chance of having a normal child. It's just the opposite of autosomal recessive. Okay? Two unaffected parents will not have an affected child. That's this one right here. Two unaffected parents would be little a, crossed with little a, and all of their children would be unaffected, right? Both males and females are affected with equal frequency because it's on the autosome. Okay, let's look at, guys, let me just warn you, these are bad. Autosomal dominant disorders are bad. Okay? Osteogenesis imperfecta, anybody ever heard of it? No? It's a collagen issue. Collagen is the substance that gives you really strong bones. There's 9 types of the disorder. Causes weak, brittle bones, mutations in 2 genes necessary to synthesize collagen, 1 in 5,000 births. And again, the 9 types range in severity. I had a student one time that worked in the neonatal unit of UAB. Now, think about that for a minute. That's where every baby, I think probably in the southeast, at least in Alabama, that is born with major problems is sent. Okay? She said that they had a baby one time come in that had this so bad. It had just been born. And it had it so bad that when they lifted up the baby to change the diaper, the bones would break. And she said they got to the point where they just didn't change the diaper anymore. And she said it only lived like 2 or 3 days. But she said it was the worst case of this she had ever seen. All racial groups. Hereditary spherocytosis, it causes fragile red blood cells. It's a blood disorder, a defective copy of a gene on chromosome 8. The cells are fragile, they're not shaped correctly, they burst easily. Anytime you have problems like that, you're going to end up with spleen issues. The spleen is where the components of the red blood cells are recycled, so these people have lots of spleen problems. Huntington disease is a neurological disorder. It's on chromosome number 4. How many people have heard of this? Huntington's? Raise your hand high on the count. 1, 2, wow, 3, 4, 5, 6, 7, 8, 9, 10, 11 people in class 12 of about 38. Does anybody know somebody with Huntington's? Want to share anything? Has it? How old? 13. Really? That's young for Huntington's. Usually Huntington's doesn't show up till middle age. His mom's got it too? Okay. Progressive degeneration of brain cells. It means the brain is degenerating. Think about what that means, guys. It starts with muscle spasms and personality kind of disorders. It usually takes 10 to 12 years after the onset of symptoms for a death to occur. This would be very similar to something like Lou Gehrig's, where you lose total control of all bodily functions at the same time, except with Lou Gehrig's, the people still have their mind. These people are also going crazy at the same time. So very, very sad. Usually, and I don't know that particular case, but usually the symptoms don't show up till middle age, 40-ish. So these people usually pass away in their early 50s. And again, this is autosomal dominant. So usually, and again, these are mutations. So mutation can happen anytime. So it could be spontaneous. But usually, you inherit this from someone who has had Huntington's disease. So think about that for just a moment. If you were a child of a Huntington parent, would you want to know if you had the allele? You wouldn't want to know? I think I would, especially before I reproduced. I mean, because basically by knowing, you're knowing that at age 40 you're going to start developing these kinds of symptoms. I worked with a banker from Coleman who was a little bit younger than me, that actually his mother had it, and he had inherited the allele, and I watched him develop this in his early 40s and then pass away. It was very, very, very sad. Okay, other autosomal dominant disorders, neurofibromatosis. These are benign tumors on nerves. Again, they're benign usually, although some people I've talked to, they have turned cancerous. They're usually benign. Think about what I just said, though. It's a tumor on your nerve. Think about how painful that would be. Some of these are so bad. I've actually seen and talked with people who had these tumors so bad as a part of this that they were disfiguring. They can actually cause bones to change shape. Yes. You can have them taken off, but they just come back, so most people choose not to have them taken off. Again, no cure. They thought, at one point, this was what the Elephant Man had. Y'all remember the Elephant Man? Ever heard of John Merrick? Except we now know he had, I think they exhumed him, and he had Proteus syndrome, which is something very similar. Marfan syndrome we've talked about. Chondroplasia is a type of dwarfism. There's lots of types of dwarfism. This one is autosomal dominant. Okay. Any questions? We're all. We've said humans have 22 pairs of chromosomes, right? 22 pairs of autosomes and 1 pair of sex chromosomes, right? Okay. So now we're going to talk about those disorders that are on the X. And specifically, we're just going to talk about the recessive ones, okay? Males, we said, were XY, and females are XX, right? We learned this several weeks ago. And we also said that the X chromosome, that we said the X carries genes for things other than gender. And I said every human has to have an X, because there are genes on the X chromosome that deal with being a human, not just a female. Those genes we call X-linked, okay? So when we're talking about traits that are on the X chromosome, we refer to them as X-linked traits, okay? We have to look at the inheritance of these differently. We can't think like the other two, because the pattern of inheritance for X-linked traits is different. Why? Because Y doesn't carry the same genes. Men only have one X. Women have 2. So I want you to think about that for a moment, okay? When we're crossing a female with a male, that's usually how it happens. Okay? And you do the Punnett square, okay? We're going to put the female alleles there, the two X's at the top of the Punnett square, the male alleles to the right of the Punnett square, and when we do the cross, we have a 50-50 chance of having a male or female child, right? You guys knew that. That's how you get that, okay? But one thing, the reason I put that on the board is because I want you to look at something. From this cross, I want you to look at the offspring that are males. Okay, take a look at the offspring that are males, right? The genes that are X-linked, okay, where would those male offspring get their alleles for those genes? Always from mom, right? Because dad is the reason the male offspring are males, because dad gave the male offspring his Y. Had dad given his offspring an X, he would be a she, right? Okay? So males always get their only allele for an X-linked trait from their mother, which causes the pattern of inheritance to be different. Male pattern baldness is X-linked. So guys, if you start developing male pattern baldness, don't look at your dad. Who gave it to you? Dear old mom's causing you to be bald, yes. Okay? Do what? It's always mama's fault. That's exactly right, okay? The pattern of inheritance for these traits that are X-linked is going to be different because males only have one allele, okay? The Y chromosome is blank for these alleles. Okay, this was discovered back in the early 1900s at Columbia University. A very, very famous scientist, Morgan, Thomas Hunt Morgan, was doing some genetic research, okay? He was working with these little fruit flies called Drosophila melanogaster. Anybody ever heard of them? You've heard of fruit flies? These are the little flies that show up when your bananas start rotting. The little bitty tiny flies that you go, where did these come from? You know, how'd they know my bananas were rotten? Because they just come out of nowhere. You know, they're these tiny, tiny little flies, Drosophila. Drosophila only have 8 chromosomes. They have 3 pairs of autosomes and 1 pair of sex chromosomes, okay? Well, he was doing crosses. He was crossing a wide-eyed male with a red-eyed female, as you see there. And think about it for a moment, okay? If this were traditional Mendelian genetics, okay, and you're crossing red with white, well, not blending, okay? Remember what Mendel's F1 was? What was Mendel's F1? If you crossed red and white, what would you expect? 100% all red. Yeah, if red was dominant over white, you would expect all of the offspring to be red-eyed. And that's exactly what he got. His F1 were all red-eyed, okay? So, I mean, he's thinking this is classic Mendelian genetics, okay? And then he crosses 2 of his F1s, okay? So if this were Mendelian, you would expect these guys to be heterozygotes, right? And if you cross 2 F1s, what are you supposed to get? Huh? Ratio should be, guys, y'all need to know this, huh? 3 to 1, red to white. Yep, your F2 should have been 3 to 1, red to white. That's exactly right. Well, he got 3 to 1, but something really weird happened. In his 3 to 1 of his F2, all of the white-eyed flies were males. And that was really strange. That shouldn't have happened. Okay? Well, in his research, he discovered that the gene locus for eye color in Drosophila is on the X chromosome. So the males were inheriting their eye color from their mothers. Okay? Okay, so let's think about that. Females, first of all, let me back up and say that when we talk about X-linked traits, we have to link it to the X chromosome, just because the pattern of inheritance is different, okay? For females, they have two X's. So you could have a homozygous red, a homozygous white, or a heterozygote female, right? Because you got two alleles. Males only have one allele. They're called hemizygous, okay? Hemi means half, like a hemisphere, okay? So males are hemizygous. They only have one allele that they inherit from their mother that determines their eye color. Okay, so let's look at what the genotypes for these flies would be, okay? A female could have two dominant alleles for eye color. That would be X big R, X big R. And again, if the trait is on the X chromosome, you have to put it there. In your problems, any problem you do with an X-linked trait has to show it on the X chromosome. So the female's genotype would be X big R, X big R, okay? If she's a heterozygote, it would be X big R, X little r. She would have red eyes. If she was a homozygous recessive, it would be X little r, X little r. Okay, other than being on the X chromosome, that looks pretty normal, doesn't it? Okay? But males only have one allele. So he only has two genotypes. He's either X big RY and has red eyes, or X little ry and has white eyes. Okay, so if we look at Morgan's crosses, that was his P generation. He had a white-eyed male and a red-eyed female. His era was thinking that white-eyed male was homozygous, okay? That white-eyed male doesn't have two recessive alleles. He only has one recessive allele because he's a he, right? So when they did the cross, they got red-eyed male and a heterozygous female, okay? Well, when he crossed two of these, his F1s, this was his result right there. And you see at the top of the Punnett square there, you see the female's alleles, X big R here, X little r there, from right there. Here's my male alleles, X big R, right there, Y there. And you do the cross and you see that all of the white-eyed flies are male, which is how he got his results. Okay, some X-linked recessive pedigree characteristics. Here's your key. This is exactly what we just went over. Again, note right here, as you're looking at the key, males who are affected only have to have one recessive allele. Females have to have two recessive alleles. Okay? So make note of that right there. Okay, let's take a look at our pedigree. And I think this pedigree is showing the passage of color blindness, I think. We have a normal female in the first generation who has two dominant alleles, mating with an affected male. Okay, to be an affected male, you only have to have one recessive allele. They produced a daughter who is a carrier. Always remember, if a male is affected, all of her daughters will get that allele. All of his daughters, I'm sorry. If a male is affected because dad gives his daughters his X, right? He only has one X. It's affected. So all of his daughters will get that allele, okay? And they have a normal son. Carrier daughter mates with normal male. They have a normal male son. A normal daughter who's a homozygous dominant, a heterozygote daughter, and an affected son. Excuse me. The normal son mated with an affected female. To be affected as a female, both of your X's have to have the affected allele. And they have an affected son. Okay? Let's look at the characteristics. More males than females are affected. When you are looking at pedigrees, that is a classic characteristic of X-linked. If you notice that there are more males affected with a disorder than females, most of the time it's going to be X-linked. Okay? because males only have one X. They can't hide it. An affected son can have parents who have the normal phenotype. Here's your affected son right there. He's getting it from his mom, who is a carrier. For a female to have the characteristic, her father must also have it, her mother must have it or be a carrier. Now that's a lot of words. Let's make sure we understand that. For a female to have it, look at that female right there, and think about where she got her alleles, okay? This female had to get one of her X's from dad, right? Well, he only has one X, so that means he's got it, right? She had to get the other X from mom, so that means either mom's a carrier or she has it, right? This female has to get an X from both parents, so dad has it, mom either has it or is a carrier, okay? The characteristic often skips a generation from grandfather to grandson. That's this right here. Grandfather through the daughter to the grandson. That's classic X-linked inheritance. If a woman has the characteristic, that's this lady right here, all of her sons will have it. A woman always gives her X to her son, his only X. If she has the disorder, all of her sons are going to have it. Any questions? Okay, let's look at some disorders that are inherited. X-linked recessive. Everybody good? Okay, color blindness. Red-green color blindness specifically. There's several different types of color blindness. Genes for red-green color receptors are located on the X chromosome. Females have this red-green color blindness if both of their X's are defective. Males if their only X is defective. We think that approximately 8% of Caucasian males have this. Okay. They actually see green as tan, they see olive green as brown, and they see red as brownish red. Okay? Does anybody know if they're colorblind? You have a friend that's colorblind? Your husband's colorblind? Really? Tell me what you see. 58, 18. Does anybody see an E here? Now you see an E? Yeah. That's actually testing your sensitivity to those particular shades. If you don't see the E, then you may have some sensitivity. You can Google this, do colorblind test. We used to have to do plates. We had them in a little notebook, and we would do colorblind tests for students if they wanted us to. But now you can Google it, and there's all kinds of these on the internet if you wanted to pursue your sensitivity to those pigments further. But isn't that interesting? Yeah. Everybody see it? Guys, I'm not lying. Several years ago, before we moved in this building, actually, I threw that up on the screen and said, "What do you see?" And a guy in the back said, "Where?" He did not know he was colorblind. He was 18 years old, and he did not know he was colorblind. I almost found it hard to believe, but he was as sincere. I mean, he did not know. Then I had another guy that admitted knowing that he was colorblind, and he started talking with the class. I love it when students share. It makes it so real when people share information about these disorders. He said he was colorblind, and another student started asking him questions, and it came, it's red-green, y'all, okay? And said, "What do you do at red lights?" And you know what he said? He said, "I usually just wait for somebody else to go." That made me not want to drive anywhere. You can see the light bulb. Yeah. Everything's gray. If he's colorblind, how does he know it's gray?
I don't know. But he was painting something, he thought it was purple, and he was like, this is purple, right? It was some kind of barn symbol, it was brown. He was like, oh, it still looks
How
good.
sad. Why would he paint the barn purple? Okay, muscular dystrophy. Muscular dystrophy is the absence of a protein called dystrophin. The absence of this protein actually allows calcium to leak into muscle cells, which causes them to be very, very weak. You have a very low muscle tone. 1 in 3,600 male births. It appears, you usually don't know it till your child starts trying to stand up to learn to walk. Gradual progressive muscle loss usually ends up with this posture right here, very young. That's a very old number right there. That's an old statistic. These people are living much longer now. Anybody know somebody with muscular dystrophy? No? One? Are they 2? How old? 7? How are they doing? He can't walk. Affected males are rarely fathers. Think about that for a moment. If affected males are not passing this to their offspring, why is it still in our population? Mothers are carriers, yes. Okay. Hemophilia is a lack of a clotting factor. 1 in 10,000 males, sometimes called bleeder's disease. It's where your blood doesn't clot. Does anybody know somebody with hemophilia? No? This is what was prevalent among the European royals at the turn of the century before last. Some of you may remember Queen Victoria and Prince Albert, Queen Victoria and Prince Albert up there. Victoria was actually a carrier for hemophilia. She passed it to several of her children. Leopold actually had it and died very young. Anyway, several of her daughters had the allele. And this was, again, ages ago. So back then, royals only married royals. Okay? So if you're a royal, your mating pool would be the royalty of other nations in Europe. So her allele literally was passed throughout Europe. It went to Spain, it went to Prussia. Probably the most famous of her alleles ended up right here. Tsar Nicholas II, and you guys remember this from history. This would be the Bolshevik Revolution. Remember, they had a son that had hemophilia that they called Rasputin. Remember Rasputin, the doctor? No. Anastasia? Come on, guys. The cartoon? Okay. Do what? You just did this? Okay. Well, now you know the science behind the son's hemophilia right there. But anyway, that allele came from Queen Victoria. Menkes syndrome is sometimes called kinky hair syndrome. It's the inability to metabolize copper. Copper is a trace element that we have to have a very, very small amount of. We have to have it, okay? But too much of it is toxic. Well, these people aren't able to metabolize, to move it. So when it enters the cell, they can't metabolize it. It accumulates, and it literally causes them to die of copper toxicity. Those symptoms right there are classic copper poisoning. These children usually don't survive past a few years. ALD, adrenoleukodystrophy, is a faulty carrier protein. It's supposed to be moving an enzyme into a peroxisome, and it doesn't, so this builds up. It causes nervous system damage. Individuals usually don't live past a young age. Some of you may remember Lorenzo's Oil back in '92, starring Nick Nolte. Okay, it was a popular movie back then. It was about a couple who had a child with this and their search for a cure. And that's it.